Thank you. It's a pleasure to be here. Thanks, organizer, for the invitation. So I will try to go through some of the examples from our work on cancer genomics, proteomics, and some of the experiences, and hopefully steer some of the kind of questions for the discussion in the afternoon. So on the kind of vertical side, we can definitely go through computational biology or integrative computational biology, cancer informatics, through integrating diverse, distributed, and heterogeneous data sets so we can build different pipelines for analysis of individual types of data. And then through different kind of networks, physical protein interactions, transcriptional network, microRNA to gene network, drug target networks, and so on, link them in order to identify potentially interesting models that we can computationally build, validate, and plan future experiments for validation or for understanding the biology of it. On the horizontal side, we can take a more kind of personalized medicine or individualized precision medicine approach where we take different kind of clinical factors, different molecular measurements, and also lifestyle or other markers, and combine them through different kind of machine learning and knowledge discovery platforms to identify different subgroups, not only of the markers, but then also of the phenotypes or patients. So we can, for example, identify or distinguish patients with a high risk of disease, low risk of disease, response, potential response to a treatment, and so on. So kind of different kind of stratification through those different integrations. The challenge is obviously we don't know which combination of these will be important. We don't know where is the noise coming from. There is different kind of missing values. There is different kind of noise and so on that I will explore later on or kind of highlight. Yesterday, there were already several talks that were discussing various challenges that we have in this kind of endeavor, which goes from heterogeneity, both in terms of time and space of the samples, intra and intratumor heterogeneities. There are different biases in terms of the data collection, different errors that come into play. But I will also try to focus on some of the aspects of where we have ignorance, where we try to build models based on incorrect data, and how can we find that something is not correct and potentially improve on it. Obviously, as we are entering this data-driven medicine and biology, a lot of the biological information turns into the numbers. Once we have numbers, there's all sorts of statistical errors, mistakes, and so on that we can do or hide certain mistakes and errors. And obviously, history taught us that there is various ways how we can have a scientific argument. We can burn witches, or as we had, for example, with Edison and Nikola Tesla argument about the AC current, electrocuting large animals to display how AC current is dangerous to humankind. And obviously, we know where the history is. So sometimes people go into extraordinary length in order to prove somebody else wrong, and only history will show otherwise. So some of these open challenges is how can we find different biomarkers and signatures, and how can we be sure that it's a relatively complete set of those markers? How can we then build models so we can understand the disease mechanism and progression, but also, for example, drug mechanism of action? Enrichment analysis obviously depends on what are we analyzing and what kind of data we are using for this kind of analysis, and I will highlight some of those things. And through that, definitely networks and so-called pathways are essential in order to help us to understand really what's the relationship between the biology and the disease. But we have to deal with different false positives and false negatives. What is the context when they are relevant? What is the coverage? And importantly also, what's the ground truth that we are comparing any of these machine learning or data mining algorithms to? So how can we successfully analyze false positives and negatives? So as a kind of starting example, what we are looking at is some of the data from TCGA, squamous cell lung adenocarcinoma. And it's basically integrating through the network everything that at that time we knew about lung cancer, squamous cell carcinoma. So it's integrating microRNA, gene expression, sequencing data, mutation, and also integrating it through the physical protein interactions, also microRNA to gene targeting networks, and also integrating on top of it what we knew about what are the prognostic genes in lung cancer, what are the significantly up and down regulated genes within lung cancer, and so on. So these kind of figures or integration not only enables you to interpret the results from these large collections of data sets across different types of high throughput platforms, but also generate models of what potentially really is important through, for example, identifying shortest path or strongly connected components within these networks. In order to do that, as yesterday, for example, Ron Shamir was showing, we can integrate large volume of this data from geo or other publicly available resources or from literature. However, garbage in, garbage out. So as pointed yesterday, we had to do curation in order to trust the information that we have. So for that purpose, we started a data integration portal focusing on some of the cancers and later on some other diseases. And in principle, you can view it as kind of we try to collect everything and anything on those diseases across different conditions, response to treatment, mutation, expression profiles, and so on. So we can view it that the rows in the large database will be individual genes, and columns in the table will be different types of analysis that will identify specific genes to be significantly deregulated under certain conditions. However, what is critically important is that we collect also clinical information that goes with it. Sometimes it might be easier than in other times. Surprisingly, there are many gene expression profiles where it will be hard-pressed to find quality information about the survival, or in lung cancer, smoking history, or RAS or EGFR mutation status. And if you don't have such information, you may have the gene expression or other profiles, but it's going to be useless for clinical application. The other thing, though, I want to highlight is for machine learning, we want to train the model on one data set and ideally independently validate on different data sets, independent data sets. However, as highlighted in lung cancer, we have to be careful because we might have independent data sets, independent PubMed IDs, yet the same patient samples are sometimes cross-used across these studies. As highlighted, we have over 6,000 patient sample profiles, but only about 4,500 are unique. So this problem is quite substantial, and we have to be careful because it can lead to significant errors, as I will show later on, or overestimation of performance. As I was mentioning earlier, for example, as surprising as it is, relatively small fraction of lung cancer data sets have information about the smoking status or detailed information about smoking status. And from other studies, we already know that it's very critical to know if it's a former smoker, but what it means, that they quit 3 weeks ago or 3 years ago or 30 years ago. As shown here in this study, there are very significant changes on a microRNA deregulation level depending on the status of the smoking, and that leads to different regulation of genes across or specific genes across those different subgroups. And it has a severe implications on survival of these patients. So all of this information is critical to understand. As I was mentioning earlier, it's critical to build model on one data set and independent validation. As shown in ovarian cancer example a few years back, perfect separation of patients in one data set, training data set, but completely missing this separation on an independent validation data set. So we try to use information of these deregulated genes with new approach of combining information from the networks in order to identify better prognostic groups of genes. So we took a set of genes that were highly deregulated in ovarian cancer across a large number of samples. But then, so it's kind of shrinking down the set of interesting genes. But then we expanded the set of interesting genes based on what is potentially related to their biology. So we started to look into their interacting partners. And then we used a relatively straightforward lasso penalized co-regression model in order to identify a smaller set of 14 genes that were prognostic for ovarian cancer. Going into independent validation, it was quite promising. So we were following up further with it. And using data from GeneSeqDB from Broad that has collection of prognostic signatures across multiple diseases, including ovarian cancer, there were 28 prognostic signatures that were published before. And we wanted to evaluate them de novo based on a hazard ratio on two large ovarian cancer data sets, Tothil and Dressman. And these numbers are basically related to PubMed IDs. So what you can see, our signature validated quite favorably compared to the other signatures on both of these data sets, but there was one signature that was basically twice as good as any other signature on the Dressman data set, but one of the poorest on a Tothil data set. So as soon as you see this kind of strong difference in terms of how it validates across two independent data sets, it kind of begged the question why. And what was interesting is that 30 patient samples that were used for training this model were actually overlapping with the Dressman data set. Again, proving the point that this is it's training and testing on a partially overlapping data set. In our case, we then switched gears. Signature was based on mRNA expression levels, and we were looking for good antibodies for the signature. We found 8 of those, and we validated the signature now on a protein level on completely independent cohort with a different method. And it validated both in terms of the prevalence and also intensity of expression of these individual proteins. So now, combining deregulated genes with their interacting partners proved very interesting, because only 2 of the genes in the signature were actually strongly differential genes. Everything else came from the network, meaning not strongly overlapping genes, differentially expressed genes. The other aspect that I wanted to highlight is there are multiple different signatures looking at the protein interaction network, and as you can see, they don't really overlap highly, because each of these studies identified one signature, and because there are a large number of possible signatures, the individual genes from those signatures may have clinical validity, but they don't overlap across. This is very much kind of recapitulating what a few years back we identified in lung cancer, where individual prognostic signatures actually overlap in a very small way. And many other people also show that one of the reasons is that we would need to identify thousands of signatures before we would start to see some kind of convergence of these individual genes that form those signatures. And in one of the papers, we actually pushed it further, and we did near exhaustive identifications of 16 signatures and identified almost 2,000 prognostically interesting signatures that were comprised of 6 genes each. Now, what was really intriguing from analyzing that set of genes, that we identified so-called best supporting actors, because a large fraction of genes were not significantly differentially expressed, but they were significantly contributing to those signatures being clinically relevant or valid. And those signatures validated on multiple independent data sets and combined data sets. So another kind of aspect to kind of highlight is, as I alluded to further, is later on we identify 15 gene prognostic and predictive signature. And as, again, same as I showed already twice, it doesn't overlap with previous validated clinical signatures in lung cancer. But through the protein interaction network, we can link it. So we can find relevance or linkages through these signaling cascades represented as protein interaction network. But as you may be able to see, some of the proteins are strongly linked and have many different connections, and others are orphans, or they don't have interactions, not because they don't interact, but because we don't have those interactions. Validity of that signature was quite substantial, because we were able to show on independent validation again that we can completely change the treatment of non-small cell lung cancer, because adding adjuvant chemotherapy significantly improved or reversed high-risk patients into more survivals. And the success story was that Helomics from Pittsburgh now markets this as a GeneFX lung prognostic gene set. Once we have these signatures, I mentioned that we can also try to understand how drug works or potentially identify different combination therapies. So we took combination of lung cancer signatures and used the CMAP data to identify ranked individual genes that will be potentially useful for treating lung cancer based on what we know about the deregulation of these individual genes in these signatures. Through that process, we identify about 250 drugs. And subset of those, we then were able to validate retrospectively on NCI60 dataset, where on all of the 9 lung cancer cell lines, they actually have stronger inhibition than the other drugs that were available. Now, it validated our approach, because we de novo were predicting which drugs might be potentially useful for lung cancer. But in terms of the lung cancer field, nothing shattering, because you can go to NCI60 and you would get the same information. So what we did was we took these 23 drugs that now validated, we took their structure, and identified 11 other drugs that were not tested on NCI60, but we predicted them as potentially useful, that were structurally similar. And what is interesting is that even on PubMed, there is very little discussion about the using of those drugs for neoplasms or treating cancer. And long story short, on 4 lung cancer cell lines, we were able to show that the top-ranking drug from that set was able to inhibit growth. And more importantly, it's not only the phenotype that we were looking for, but we were also able to identify in all those 4 different cell lines that the genes that we predicted should be reversed as after the transcriptionally reversed after the application of the drugs. They did significantly change. So it's not only identifying the phenotype, but also having a mechanism. How does that work? So going back to the signatures, I highlighted that there are multiple signatures that we can identify, but we don't have computational power to find them all. So we started a project with IBM World Community Grid mapping cancer markers to try to kind of push for identifying stronger these signatures. And as I have highlighted before, two aspects to be remembered. One is if we take univariate analysis and compare to the signature, signature beats any single gene most of the time, all of the time. That's one part of the message. But the other part of the message is that the second worst gene on an individual basis is actually the third best gene when we have a signature. So that's an important aspect for these best supporting actors, that something that is ridiculously unusable, and we would most likely filter it out before we will be trying to search for the best signature, we would filter it out before we will have a chance of identifying it. What is really intriguing is that this recapitulates to different sizes of signatures. So this is univariate versus signature, but when we compare signature of different size, 10 and 40 genes or probes, same order of probes on both graphs, and the height of individual bars shows how important is that gene contributing to the signature of a specific size. And we can see that a gene that is mediocre in a 10-gene signature is now best marker in a 40-gene signature. So finding the good signature obviously is a factor of how big signature are we looking for. And there are many different aspects that we have to consider. One is larger signatures have a broader applicability across more heterogeneous populations of patients, but obviously smaller signature is more clinically plausible in terms of the cost, noise, and so on. So one has to be careful how we optimize these two parts, and the jury is still out there what will be necessary. Without going into, the project is still going, but without going into too much detail, I want to highlight again the aspect that the most interesting genes now, that includes differential genes and the best supporting actors from large-scale analysis on a World Community Grid, also shows that some of them we have a large number of information about who they are interacting with, and for many of them we have absolutely no information who they are interacting with. So interactomorphons, we cannot build models, we cannot understand how they link to signaling cascades. And that brings us to another example of why type of data that we use for these protein interactions and pathways is essential, because it may or may not enable us to form certain hypotheses. In a different study, we identified 20 gene prognostic signatures from surrounding stroma in lung cancer. And I want to highlight the aspect that because a lot of these analyses use enrichment analysis, use mapping to the protein interaction network, we study only a small fraction of the genome that we have well characterized on the interaction network and ignore everything else. Proof to the story, using interaction database that integrates multiple curated and high throughput databases, we can take the 20 genes, which are the square on the outside, and identify how they link on a protein interaction level. What is not surprising that they don't interact directly. If they would interact directly, likely they will be redundant and we would have 19 or 18 gene signature, not 20. What is important, though, that they strongly link to this middle layer of proteins that interconnect multiple proteins from the signature. Those are the red edges. And those proteins are highly interconnected into highly connected components, which are the blue edges. So we now have a very interesting potential story of how those 20 genes relate to the biology, to the signaling cascade, and so on. Using exactly the same analysis, but only one of the databases, HBRD, for example, clearly shows you that there is plenty of proteins that have absolutely no connection. So we cannot even start making any discussion about what potentially relates among these individual proteins. So skeptics may argue that obviously there could be noise here because we are integrating multiple types of data, but we have a very good clinical context that enable us to validate specific interactions if they prove to be useful. And it's better to have information that we can validate as opposed to have absolutely no information and blindly sit and not do anything. Obviously, as we integrate more data, the network starts to be more dense. And so we had to start kind of thinking about the scalability of graph theoretical algorithms that can go into and detangle these large networks. But we still need more of those protein interactions, because at least 10% of the protein interactions remains unknown, and we have to analyze them because they relate to multiple different diseases, regardless of what we look at, either census mutations or OMIM disease database or our cancer data integration portal, zero or low degree proteins are strongly linked to multiple diseases across these profiles. So we had to really find different methods for this interaction prediction. We derived one such method, and as in traditional machine learning algorithm approach, we identify true positive protein interactions and identify those unlikely interactions as a starting point for a training set, and then describe those both sets in terms of different features and the combination of features, feature sets, from gene ontology, from sequence information, expression profiles, and so on. Then each of the pairs of potential pairs of interacting partners were further identified through co-expression, expression through different tissues, homologs, orthologs, and also protein interaction data. When we combine all of these different pieces of evidence through noisy OR, we can identify probability with evidence from individual sets and feature sets that can identify, should be time.
5 minutes.
No? We started later. But okay.
You should have some time for questions.
So what is interesting about the integration is that individual features, like, for example, topology from the network, is useful up to a certain point, and then it becomes useless because we are in a very sparse or completely disconnected part of the network. And what is important is that when we combine those pieces of evidence, even at a time when we have very little information available from single features, the combined features can still actually provide reasonable prediction accuracy. To validate it, we looked into multiple different ways of comparing to existing prediction algorithms, but also to high-throughput assays. As, for example, in Vidal's group, there were 5 high-throughput assays compared on true positives and true negatives. All of the green blocks basically identify true positive interactions, and these are the false discovery rates. What is important is that none of these 5 methods, but there were no mass spec, for example, they were only run once, and so on. So there are many technical reasons for why they miss 41% of the interactions. But what is important is that the computational prediction identifies 91% of interactions with only 1% false discovery rate. So it quite favorably compares to high-throughput method, quite comparable to, or much better in terms of the false discovery rate and sensitivity to other prediction algorithms. And also through very extensive 3 different way validation across novel mammoth screen, GST pool now, and co-IP, significantly increasing the validation rates compared to biological assays. So combination of computational predictions with biological validation significantly improves sensitivity of these assays, and we can go after novel targets. One of the such example was EGFR, very important protein in receptor protein in lung cancer. And the idea was not only to identify and validate novel interactions for EGFR, but also then look at the wild type versus specific mutant L858R, which is frequent in lung cancer, how mutation affects interacting partners. So all of these dots in here are basically validated protein interactions. But what you can identify is that those that are more on the upper side are specific to mutant version of EGFR, whereas those on the lower side are specific to the wild type version. When you look into these individual dots, most of them are triangles, either pointing up or down, and that's from the cancer data integration portal if we know that those genes are up or down regulated in lung cancer. And those that are highlighted with red are prognostic genes in lung cancer. So it's not surprising, most of the interacting partners of EGFR are related or deregulated in lung cancer. But long story short, we picked one of those, CRK. And what was interesting is when we combine it with shRNA screen, we're able to identify that knocking it down increases erlotinib sensitivity and validating it at least on a cell line, that we can combine deregulated genes, signatures, protein interactions, predictions, and other type of data in order to identify potentially novel set of patients that can be treated with available treatment strategy. These interactions are really essential because, as mentioned earlier, we can combine novel high-throughput screen with novel predictions, and only then we can actually understand how and why drugs work. So this is highlighting drug targets, and this is highlighting genes that we know are deregulated from transcriptional profiles by those drugs. And as highlighted here, novel myth screen and interactions through I2D. It's the combination of those interactions that we need to have in order to understand what's the linkage between the drug target and genes that are transcriptionally modulated by a specific drug. So considering that this is really useful, we switched gears and integrated other prediction algorithms and now extended number of interactions for human, for example, to over 800,000 interactions and covering almost the full proteome as we know it, and also then going through 5 other model organisms. What is important that we can then annotate mostly for human and mouse, but also sporadically for other organisms when available, was the expression of these targets and these interactions and conservation of these interactions across different species and across different tissues. And thus potentially starting to detangle not only that, okay, we have a putative 800,000 interaction network for human, but in which tissue, under what condition, in which disease, these interactions may be phenotypically interesting or useful. So in order to be roughly on time, I will skip through a few other examples and I will jump to the end. And one of the examples I didn't have time to go into, but it's related to the aspect of we have large number of information on molecular profiles and some information on the clinical information about patient. And what is amazing is you can open any journal these days, and if there is a clinical study, people will refer to BMI, body mass index. And what is shocking is that in this day of age, where we have other alternatives for several months and years available, it's really useless, because when we look into Schwarzenegger and Danny DeVito, they have pretty much the same BMI. And nobody will convince me that in terms of the health and fitness level, they would be anywhere to be comparable. So we will try to compare molecular profiles and identify what are the risk factors for individual patients based on a BMI. It's completely useless. It's like using a sand clock and a digital clock at the same time. So what is important, we have to use different methods, and there are methods available, for example, for BMI. We can use data fusion in order to reduce some of the false positives, and through provenance, we can also ensure that we can control what false positives and false negatives. And through integrative analysis, we will be able to refine evidence-based medicine, because there are many, many errors in the literature. And again, one of the aspects that I skipped through is, I don't know how many people looked into increasing amount of retracted papers in the literature. Last year, it was about 5% of the papers were retracted, and it's exponentially increasing over the last few years. And many times when you find a discovery or we create a model, we find some interesting findings, and we have a trouble of convincing, it was also discussed yesterday, convincing reviewers what is right, what is wrong, because there is a published literature that shows otherwise. And in these studies, it actually shows that a large fraction of these papers will be eventually retracted. Most of these papers are clinical, and thus we have to really be rigorous in terms of how we prove or disprove what is true, what is right. I would like to acknowledge a lot of collaborators across different diseases. Some of them I didn't mention here, but obviously it's collective work that led to some of these findings. And I highlighted some of the people involved in the lab, but obviously programmers that enable that these servers are up and running are essential part of the lab. And if people are interested, servers are available with this data at different parts, and I will be happy to take any questions.
Well, I can start with the immediate question. Is there any real computational challenge in figuring out duplicates in data? And yeah, the IDs may be different, but can't you just tell immediately by looking at the data set? Or is it going to be slightly different here and there?
Slightly different. One aspect might be that there will be different platform that was used, for example. So you cannot kind of really find a carbon copy of the data. It's just, I mean, sometimes you really had to go into supplemental Excel spreadsheets to identify patient ID that eventually you trace it back that is the same sample. I mean, the sample from the same patient that is used in multiple different data sets.
And the technology issue is an essential issue, obviously, in replicating your results. So maybe the key question here is, in order to, is it really an integration issue, or is it just making sure that the kind of data that we look at is comparing apples and apples and nothing more? Just.
So I take it, at different steps. At one step in terms of the curation, it's really ensuring that we curate and clean the data before we put it into the pipelines. The next aspect is that once we start integrating, call it different pipelines, into identifying more cross-platform, cross-entity measurements, that's where the networks come in. And obviously it depends on what networks are we using. In a sense, is it reducing the error or noise, or is it increasing it? And what's the relevance? Obviously, at one point, when everything will be connected to everything, it's not going to be useful, right? So that's also one of the reasons why we still need more of the interactions. We still need more of the microRNA to gene targets. But we also now really strongly will need to contextualize these networks, because at one level, as I said, everything will be connected to everything, but the question is, in lung cancer, when we have EGFR mutation, which portion of this network is relevant? In ovarian cancer, when we have mucinous subtype, different configuration of this network might be relevant. So it's going to be contextualizing it and then analyzing that contextualized version or instance of a network.
So what exactly is the difference between your signature and, say, a bag of genes or proteins? I just wasn't clear.
So signature, in my definition, is that it has either prognostic or predictive value. So either you can identify patients with a high or low risk, or you can identify, for example, which patients will or will not respond to a specific treatment.
I understand the goal of the signature, but what is in the signature? Like a bunch of genes, their mRNA
or proteins,
levels,
yeah.
Protein mRNA levels, and that's it? Or like connection, are there non-additive terms in the signature?
Non-additive terms?
It's just a vector.
Pretty much.
It's just a selection of genes and protein levels.
Specific selection, but what is important is that they are not necessarily, as I was highlighting, not necessarily the most differential. So we don't really have still a good way of identifying the best signature, and as I also highlighted is that there is no such thing as this is the best signature, or this is the best set of genes, because if we are looking for a less than 10 gene signature, you can rank a few thousand of them based on, for example, hazard ratios, how you can validate them on independent data sets. But if you will be looking for, say, a 50 gene or 50 protein signature, it will be a different set of genes or proteins.
So the point you made about the best supporting actor is you include some genes that maybe in the training set they don't differ all that much, but by networks they will differ in a test set.
So the example I was showing on ovarian cancer, we started with 114 genes that were highly differential across a large set of samples. Then we expanded it to close to 600 proteins that were connecting among themselves. From that, we identified 14 genes based on independent validation and cross-validation that were now a strong prognostic signature in ovarian cancer. And what was interesting is that only 2 of those 14 genes actually came from those highly differential genes. Everything else were interconnectors from the network. So that's one of the kind of highlight, that it's through the networks that we can find clinically more interesting set of genes or proteins that can validate on independent data sets and also then validate on a protein level. And we have some, I didn't show, but we have some now validation through the mouse models of really the biology of why and how the disease progresses. And it's only because we were able to expand it through the protein interaction network. We would not find it if we would focus just on the differential genes.
So we should take the rest of the questions offline. Speaker again.